Hey there, folks, and welcome to yet another COVID literature review. If you follow me over on TikTok, this is a series that you may know of. Effectively, we hop into the seedy underbelly that is academic literature and see what's going on in the world of COVID-19. Now, I should say at the outset, I am not a medical professional. I'm not a virologist. I am just a scientifically literate person talking to a camera on the internet, so you should take what I say with an appropriate grain of salt. But at the same time, we're going to be looking at the paper together. So if you agree, disagree with things that I'm saying, feel free to hop into the comments. As you can tell, I'm still sort of building YouTube muscles here, so any feedback is sincerely welcomed. So without further ado, we're going to be looking at this paper today. It was published in the Annals of Internal Medicine. I'm just going to quickly put a card over there on the video that talks a little bit about how to decide if academic literature is worth your time, because that's surprisingly challenging depending on where you get it. I'm going to assume that you have watched that video and have context for how robust this science is. It's pretty robust, but it is a single case study, and we'll talk more about that later. So, title of the paper, Replication-Competent Virus Detected in Blood of a Fatal COVID-19 Case. What I want to say at the outset here on this phraseology, replication-competent virus, you may remember, if you think back to high school biology, there's an ongoing, I would argue, philosophical debate about whether or not viruses are alive or not, and replication-competent virus is basically a way of saying like a live virus, because if the virus can replicate, it can make copies of itself and proliferate and cause damage, and that's, as hosts, that's what we care about, practically speaking. So anyway, if we head on into the paper and take a look at the background, SARS-CoV-2, which, by the way, SARS-CoV-2 is the virus, the causative agent that causes COVID-19. So SARS-CoV-2 is a virus, much like HIV is a virus, and COVID is an illness that is caused by SARS-CoV-2, much like HIV can cause AIDS. So anyway, SARS-CoV-2 infects respiratory tissues, and in some cases, replication-competent virus has been detected in extrapulmonary tissues, including the brain. Now, this citation here links out to this Nature paper that made quite a splash when it was published. Highly encourage you to read this if you're interested in this sort of stuff. But basically, they did a series of 44 autopsies at the NIH of patients who either died from or with COVID-19. I think from is kind of the selection criteria. But anyway, they found a lot of really interesting findings in this paper. So again, it's worth looking at, but that's not what we're talking about today. Okay, so viremic spread is suspected, and detection of viral RNA in blood is frequently reported. So what they're basically saying here, in case you're unfamiliar with what this word means, viremic is vir, virus, emic, blood. So what they're basically saying here is, because we know that it gets into the lung tissue, but then we can find SARS-CoV-2 in other tissues throughout the body, the suspicion is that the mechanism for this is by the virus traveling through the blood. And they mention viral RNA in blood, and they talk a little bit about this later, but they mention this because viral RNA being found in a tissue, blood, is suggestive that there's replication going on. And that's because viral replication is a pretty messy process, and it involves the destruction of your own cells. That's how the virus replicates. And so that can lead to RNA being sort of discharged into the tissue, which in this case is blood. So again, that kind of suggests that there should be replication-competent virus in the blood, but as of yet, nobody has actually demonstrated that that's the case. So no one has ever actually found competent SARS-CoV-2 circulating in the blood, which is really quite interesting, actually. So anyway, the objective of the paper is to confirm viremia in a fatal COVID-19 case where viral sequence in blood and tissues match. So what they're saying here is it's not enough to detect SARS-CoV-2 in the blood, because that, it could have gotten there from other means. It might be a different variant. Like, we want to know for sure that the virus started in the lungs, got into the blood, and then made it to some other tissue. And this is essentially, they're doing like a DNA test to prove it's the same virus, except it's RNA. You know what I mean? So here they present the patient. I'm not going to go too into detail here. Just want to thank the family of this patient for allowing this science to be done. It means it's a sacrifice that they're making. SARS-CoV-2 was confirmed by nasopharyngeal swab using PCR. So we know that this patient had a SARS-CoV-2 infection, which probably contributed to this decompensation. Again, it looks like the patient was quite ill beforehand, but of course, we know that if you have previous conditions, it makes COVID a lot more dangerous. Anyway, moving on. They talk a little bit about how the samples were collected once the patient passed away, and then here's where we get to the actual results. SARS-CoV-2 RNA and subgenomic RNA, suggestive of recent virus replications, we were just talking about this, were detected in plasma, lung, heart, and several additional tissues. And SARS-CoV-2, the actual virus, was detected by ViroE6 cell culture in lung and heart, but not plasma as previously reported. So initially, they used this ViroE6 cell culture, which is, you can just think of it as, this is the way that they try to isolate the virus. That's the technique that they're using. And so they do this, and they find the virus in the lung and the heart, which suggests that it made it from the lungs to the heart, but it wasn't found in the plasma. So again, we haven't seen viremia, so we don't know how it got there. And now here's where they made kind of a little bit of a breakthrough. A second effort to isolate virus from plasma using a modified Viro cell line stably expressing human transmembrane protease serine 2 and angiotensin converting enzyme was successful. Now, I'm going to speculate a little bit here. I suspect that the ACE2 being expressed on the cell was what really did this, because one of the things that we know from other papers is that part of the reason why SARS-CoV-2 can cause vascular injury and can cause damage to the GI tract and the heart is because of the fact that it's very good at binding to ACE2, which is a thing that is ever present in our bodies, and a lot of our cells use it to allow stuff in and out of the cell. They're like little little tunnels that allow you in and out. It's like kind of driving into Manhattan through the Lincoln Tunnel. So anyway, by adding ACE2 to this cell, my personal, the mechanism that I have in my head is that there was just enough replication competent virus in the sample that it, knocked into the ACE2 receptor and then used its spike protein to get into the cell, and that's how they were able to detect it. Again, I'm speculating a little bit here, but we know that ACE2 is a big part of SARS-CoV-2's pathophysiology. So really interesting. If you're wondering why nobody tried this before, or why it took so long to isolate SARS-CoV-2 from blood tissue, we're actually going to get a little bit of insight into that later. But anyway, the rest of this paragraph is just talking a little bit about how the concentrations of viral RNA sort of drop off day over day. And this points to an observation that, again, has been made in other papers, and this letter actually mentions it as well. Over time, basically the period of time where there's a lot of SARS-CoV-2 RNA in your blood, which they call RNAemia, which I think is just such a classic science word, that drops off pretty quickly after the acute phase of the infection. It spikes sort of in the first week where you're symptomatic, and then it kind of drops off, which sort of makes sense if you think about it, because the innate immune response kicks into play. We know that SARS-CoV-2 is actually pretty good at avoiding the innate response, and it can actually destroy neutrophils. Maybe I'll do a video about that at some point. But anyway, it makes sense that before the immune system really has an opportunity to kind of ramp up, SARS-CoV-2 is able to get into the blood, replicate, go to distant tissues, and then once the immune system has really woken up, it gets removed from the blood. The viremia resolves. The other thing that I mentioned in the beginning or that the paper mentions in the beginning, is that they also wanted to prove that it was the same virus in the lung, blood, and cardiac, and they were able to do that. The samples, they did a full-length genomic sequence of the pre- and post-viral culture of plasma, lung, and cardiac samples, and there's 99% sequence similarity, which is pretty convincing, I think. And there were also some conserved mutations, which is really fascinating. So it's quite likely that these were indeed the same virus. Said another way, the virus that got into the lungs probably was the virus that got into the plasma that got into the heart. That's what this finding suggests. And again, they mentioned that there are 3 synonymous and 1 synonymous mutations present in 45 to 62% of reads in the post-viral culture plasma sample. So there's some irregularities, but it's important to remember that viral replication is a pretty messy process. And in particular, we know SARS-CoV-2 has the ability to kind of change the way that it looks, and the way that it does that is by mutating. So this doesn't call this finding into question, I don't think. So anyway, we mentioned earlier that it's kind of bizarre that nobody else has been able to find this up until now. And so you might be wondering, like, why is that? And the paper goes on to explain that once they had this process that they knew worked, they thought to themselves, okay, well, let's go back to this original patient report of 44 autopsies, the one that was published in Nature over here. And let's try and see if we can isolate this from any other patients. Indeed, this patient, the one that they tested, had the highest level of SARS-CoV-2 RNA. So they picked this patient because it was the most likely to have viremia, to actually be able to detect SARS-CoV-2. Now, they found that of this population of 44 patients, 12 of the patients did still have RNA in their blood. So that suggests that there was some replication-competent virus in the plasma semi-recently, historically speaking. And then they report some other data about IgM, IgG, and they redo this modified virocell test in 7 additional patients with SARS-CoV-2 anemia, ranging from this to that. Again, they're just saying they investigated some patients that also had this viral RNA, and all were negative. So this suggests a couple of things. Actually, before I opine, I'll just share with you their discussion. So they claim our case proves that replication-competent SARS-CoV-2 can traffic in blood during COVID-19 and seed tissues throughout the body. So they're saying quite definitively, and understandably so, that this case study really kind of confirms the theory that the virus can get from the lung tissue into the plasma, circulate through the body, and then infect other tissues. Now, they do also say that further studies are needed to determine the implications of our findings for persons infected with SARS-CoV-2 variants, those with mild illness. And I want to come back to this because I think it's important. Persons recently vaccinated or persons with waning immunity after natural infection or vaccination. Now, the reason why I think mild illness is important to highlight here is that one of the concerns that, I'll just say I personally have, and this is an opinion that's shared by a lot of people, is we're letting all these people get infected with SARS-CoV-2. And if you're young and you don't have any prior medical conditions, a lot of people have COVID, they're sick for, a week or two, they feel like crap, but then they get better. And, there's been a lot of research done about the long-term impacts of COVID. It's also important to remember that, COVID-19 as a disease has only really been around for 4 years. So we're still learning a lot about what the long-term consequences of a SARS-CoV-2 infection are. But as these, authors are kind of highlighting, just because a person recovers doesn't mean that there isn't an ongoing issue here with their health. Because again, if the virus can get to, distant tissues and kind of hide out there, and viral reservoir is a potential mechanism for long COVID, there might be long-term damage in patients who aren't exhibiting symptoms of long COVID. Anyway, I've strayed squarely into speculation corner, so I'm going to call the video here. Thank you for watching. If you made it all the way to the end, again, if you want to know a little bit about what makes a good academic paper, I'll post a link to that here. And again, thank you for watching. As always, comment, like, subscribe, all that stuff that makes the algorithm happy. And again, looking for feedback here. So if there's something that you liked about this video, you didn't like, I'd love to hear about it. Thanks for watching.